Our next speaker is Yuankai He from the University of Cambridge.
It is my pleasure to present this study on behalf of all the co-authors. It is a genome-wide association study of the macroscale resting state functional brain network. So, disclosures first, it is not relevant to this study. To bring everybody onto the same page, for those who did not do fMRI, the brain displays fluctuations of brain activity, even at rest, which means in absence of extrinsic stimuli. These fluctuations are spatiotemporally coordinated, and by cross-correlating different brain regions, we get the resting state functional network. To describe the topological properties of the resting state functional network, including modularity, segregation, integration, rich club structure, we can use graph theory metrics. Whilst these graph theory metrics and the resting state network as a whole have been quite extensively studied phenotypically, large gaps remain in terms of the genetic underpinnings of the resting state functional network. From what limited studies we have, we know that it is significantly heritable, but the genetic loci from what few GWAS that we have are quite heterogeneous. There have been only 2 overlaps out of like 20 loci that are identified. Another gap in the current study is that nobody has actually looked at the asymmetry of the functional network, which is actually phenotypically important as it's implicated in schizophrenia and ADHD. And also the spatial resolution in terms of the regional topology of the resting state functional network have been quite limited. So the first question we asked ourselves is, what is the underlying genetics of the resting state functional network topology at both global and local levels? On the basis of that, we asked ourselves if the brain structure actually genetically overlaps with functional network topology. Various phenotypic theories have been proposed linking structure and function, including the microstructural white matter connectome and macrostructural geometry. Both are proposed to constrain on the functional network, but from phenotypic studies, it's very difficult to infer causality, whereas in genetics, it is theoretically possible, as we'll show later. Finally, we asked ourselves, given that functional network has been associated as imaging structures with many psychiatric disorders, if they are genetically correlated with neuropsychiatric disorders. In particular, functional connectivities, abnormalities have been observed in many disorders, but these signatures are not really perfectly overlapping with structural abnormalities. So we specifically asked ourselves whether the functional genetic overlap is structure independent. To start with these studies, we obtained pre-processed functional MRI images from the UK Biobank, aligned them to the Glasser Atlas, denoised the regional time series, obtained the resting state functional network, and obtained 7 graph metrics, including degree, the ipsilateral, contralateral proportions of degree, efficiency, clustering, path length, and small wildness. For these graph measures, we first conducted GWAS at global level and identified 1 significant locus on chromosome 21. For regional phenotypes, after accounting for multiple testing, genomic regional overlap, and brain regional overlap, we identified 7 independent loci. And we also identified the regional phenotype genetic signal to be quite consistent with the global level graph phenotypes, with the largest shared genetic loci shared across 201 brain regions, but also global graph phenotypes. The genetic correlation was also generally above 70%, so very high. Next, we analyzed whether there are shared genetics between these graph phenotypes. All graph phenotypes have been identified as significantly heritable, so about 5 to 7% consistent with previous studies, whereas theory suggests that different graph metrics describe network integration, segregation, and small wildness, or the balance between integration and segregation. The individual differences between these graph metrics are quite similar, both phenotypically below the diagonal and genetically above the diagonal. On the other hand, to the bottom left corner, we see the asymmetry of the functional network as a separate cluster, also very highly correlated. Before we analyzed the overlap with structure, we took a deeper dive into the candidate genes and cell types, so potential mechanisms of the underlying the functional network organization. From 11 different methods, we prioritized 10 candidate genes, and of particular note were 4 genes that were implicated in brain development. Worth particularly mentioning is cyclin D kinase 2, which is a cell cycle regulator, and mutations in CDK2 has been previously reported to be associated with human microcephaly. So one potential line of evidence suggesting structural functional overlap. Furthermore, we conducted cell type enrichment analyses by testing these gene-level effect sizes with single-cell RNA sequencing data for top 10% expressed genes in each cell type. We identified the functional asymmetry phenotypes to be enriched in oligodendrocytes, which overlaps with previous enrichment of global microstructure or water diffusion phenotypes, also in oligodendrocytes. Another line of evidence suggesting structural functional overlap. We proceeded to formally test this structural functional overlap hypothesis by utilizing a previous GWAS study from our lab, which contained both macro and microstructure. First of all, we conducted genetic correlation between the functional and structural phenotypes. We observed quite different correlation patterns, whereas global level graph phenotypes of the functional network correlated with cortical expansion phenotypes of macrostructure, so volume and photon index. The microstructural phenotypes were correlated with asymmetry, in particular mean diffusivity, isotropic volume, which are water diffusion phenotype. Based on these significant correlations, we proceeded to infer causality from structure to function. To bring everybody onto the same page, we leveraged a statistical tool called Mendelian randomization, which is comparable to a randomized controlled trial, but instead of randomizing drugs and placebos, we were randomizing genetic variants. The hypothesis is that if a genetic or genetic variants that have a direct influence on the exposure phenotype also have a proportionate effect size onto the outcome phenotype, then a causality from the exposure to the outcome can be inferred. There are various assumptions for Mendelian randomization, for example, no confounding and no direct effect on outcome phenotype. So we use different methods to account for these violated assumptions. On these graphs, each cross represents the effect size and standard error of an independent variant on both structural and functional phenotypes. And from this graph, it is very clear that volume is causative to path length, so the top graph, whereas the causality is not observed in the reverse direction. We replicated this finding with other cortical expansion phenotype, and also worth noting is that we also inferred a causal relationship from microstructural asymmetry and not vice versa. Finally, we investigated whether there are shared genetics between network phenotypes and psychiatric conditions. Of all the psychiatric conditions we screened, there were only autism that was significantly correlated with the network phenotypes, and this correlation occurred at both global and regional levels. Further supporting this correlation was its spatial overlap with clinical imaging signatures in the frontoparietal network, which showed significantly higher correlation with graph phenotypes compared to other brain regions. We also confirmed these results with polygenic risk score of autism and also using 3 different autism GWASs. Also worth noting is that structural phenotypes, in particular cortical expansion phenotypes, were not correlated with autism. However, we did not identify any causal relationship in either direction using Mendelian randomization. So conclusions. The first aim, we identified 2 correlated clusters amongst network phenotypes, one across all the graph phenotypes, but also including regional level graph phenotypes, the other cluster including the asymmetry of the functional network. The next aim, we identified a dual constraint from both cortical macro and microstructure, which have causal effects on different aspects of functional network topology. Next, we identified a rather specific correlation or overlap between network phenotypes and autism, which calls for caution in interpreting functional imaging signatures. Finally, it's worth mentioning that this project is part of a bigger project, and it's very pleasing to see the GWAS atlas unfolding across different imaging modalities, including structure similarity network, and functional gradients, and for this study, functional graph phenotypes. Before I proceed to questions, I would like to thank my co-authors, funders. Please scan the QR code if you want to consult a free preprint. The poster is not out yet, but expect you on Friday and Saturday. Thank you very much.
Thank you for a very nice presentation. So we've got a question here. With the bias of GWAS toward rare phenotypes, will another analytic method be more suitable to uncover the genetic underpinning?
Sorry, I didn't quite understand. Which phenotypes?
Rare phenotypes, or genotypes, sorry, genotypes, not phenotypes.
Rare variant genotypes, I believe there is a rather different set of analytic tools. So I think that's our next step in our analysis to analyze the rare variant basis of different imaging phenotypes. But so far, studies into the rare variant basis of brain phenotypes have been quite limited.
Right, okay. And sorry. Can the genetic variants identified also be validated using mouse studies? So, for example, using MRI of genetic knockouts?
I believe that because this is this is a common variant GWAS. So one point of caution is that all those variants identified in here, they rather have low effect sizes. So the effects observed in translational models might not be significant. Another point of concern is that this study used HCP or Glasser atlas. I don't believe there is such a fine resolution atlas which is comparable to the human regions in mice, let alone some brain regions that are highly specific to human. So I would exercise very much caution in that regard.
Well, thank you very much for a beautiful presentation. So again, you can catch all our speakers after the session.